spiel here. Yeah, welcome to the Temporal Genomics seminar. Thank you everyone for coming. My name is Brennan Reed. I'm a postdoc in Malin Pinsky's lab at Rutgers University in New Jersey, speaking to you from the traditional territory of the Lenni Lenape people. And this series is organized by the Research Coordination Network for Evolution and Changing Seas Temporal Genomics Working Group. If you want to learn more about our working group, we have a lot of information on our website, and we have a schedule for the seminar series, and I will link that website in the chat in a little bit. But yeah, this is a bi-weekly seminar aimed at showcasing research into temporal genomics. And by temporal genomics, we mean studies that use genetic samples collected over time, often historic and contemporary genetic samples, to examine questions that are relevant to the ecology and evolution of species over the past, 10 to several hundred years, especially their responses to human-induced changes in the environment. We're also really interested in highlighting best practices and new methods for those of us who are interested in doing these kinds of temporal genomics work too. So we have a really wide range of topics and speakers planned for the seminar series. We hope that you'll stay with us over the course of the next few months. The schedule is on our website, as I said before. Also note that we don't have enough time in the seminar series to highlight all of the amazing work that people are doing in temporal genomics. So we are planning a Twitter conference where even more people will be able to showcase their research. Details are forthcoming. That's probably going to happen sometime in the summer. If you'd like to participate in that, please stay tuned. So just a few ground rules. While our speaker is presenting, we ask you to please be respectful, keep yourself muted, hold all questions until after the presentation is over. Once the speaker is done, we ask you to either use the raise hand button or indicate in the chat that you'd like to ask a question. Katrina Catalano is helping me moderate this seminar. Either she or I will call on you, and then you can ask your question. Also note we have quite a few registrants. This might be a stress test on our Zoom room. If something unforeseen happens, we do have an alternate webinar link that's available, and we will do our best to email that link out to you quickly and then resume as soon as possible in the webinar. But hopefully we won't have to do that. And all right, so I don't want to take up too much time from our speaker. So now that that's all out of the way, let's give a big welcome to this week's speaker, Dr. Shane Campbell-Staton. Shane earned his Bachelor's of Science in Ecology and Evolution at the University of Rochester. He did his PhD at Harvard, working with Jonathan Losos and Scott Edwards, where his dissertation was on phylogeographic history and temperature-mediated evolution of the green anole, Anolis carolinensis. And he went on to do some postdoctoral work as an NSF fellow at University of Illinois, Champaign-Urbana, working with Julian Ketchen. And also as a postdoc, he worked with Zach Chevron at University of Montana. He landed a faculty position at UCLA in 2018, where he's currently an assistant professor in the Department of Ecology and Evolutionary Biology, and also at the Institute for Society and Genetics. He's also a skilled and multifaceted science communicator. He teaches a class and produces a podcast called Biology of Superheroes, where in the most recent episode, he discussed the biology of Ant-Man's insect brethren with Corey Moreau of Cornell and his co-host Arian Darby. So today, Shane will be talking about some recent work that he's done on temporal genomics and biological response to anthropogenic change. So Shane, you can take the floor.
Awesome. Thank you so much. And thank you for having me. Let me share my screen here. Oh, share that. Share that. Okay, everybody can see this okay? Awesome. Yeah, it's always, it's always a little nerve-wracking, when people give the intro and they're like, oh, he's such a great science communicator, and then I have to give a talk right afterwards and then, embarrass myself. But I am really excited to share some of my work with you all today. This idea of temporal genomics, I never really considered, my work specifically in within the context of temporal genomics, although, now thinking about it, a lot of my work certainly does fit into that category. And most of my research, my lab's research, focuses on trying to understand the evolutionary impacts of our human footprint. So how humans act as engines of evolutionary change for mostly non-human species in a bunch of different ways. So I'm going to give, I'm going to talk about 3 different projects. One is actually a project I haven't talked about in quite some time. One is a project that we are actively working on right now. And then one is a project that is currently in revision. And each of these, I think, approaches this idea of temporal genomics in slightly different ways. So we're going to play around with this term temporal genomics and try to illustrate a few points using these 3 different stories. All right, so when we consider this relationship between evolution and anthropogenic change, I actually think this is a very powerful and interesting relationship because on one hand, evolution reveals the lasting biological impacts of anthropogenic change, but then on the flip side, anthropogenic change, provides unique insights about the basic and fundamental processes of evolution. And I think temporal genomics can be a very valuable and powerful tool in understanding how these 2 things speak to each other. So temporal genomics, when we're talking about temporal genomics, we're going to start off talking about temporal functional genomics, like trying to understand, like not just looking at sequence level change, but also looking at patterns of gene expression and trying to understand functional change across time in response to, in this case, extreme weather events. When we're talking about extreme weather, across the planet, as when we think about climate change, we think about global warming as well. We should sort of the prevailing trend of climate change, but you also get this uptick in both the frequency and magnitude of extreme weather events across all different axes. Things like hurricanes, droughts, even extreme winter events. And at mid latitudes, this becomes very important because there is, since the 50s, there's been this uptick in both the frequency and intensity of winter storms, extreme winter storms at mid latitudes. And some of this activity is caused by the polar vortex, which sort of sounds like a B movie, with like a bunch of polar bears and orcas in a vortex of snow going around killing people. But in actuality, it is a pretty steady pattern of Arctic air that circles around our poles. But as the planet warms, this pattern becomes more wavy and sends periodic bouts of this Arctic weather farther south, causing these extreme weather events, like this winter storm that passed through the South in January 2014. Now, I'm from the South. I was raised in South Carolina, and I can tell you, we don't do the cold. We don't like it. It doesn't like us. The 2 just don't go together. And I remember when this storm happened, I was sort of on the Boston Globe looking at the fallout of this event. People were like trapped in the Piggly Wiggly, and people like trying to ski home, and their trucks overturned in like literally like an inch of snow, but like people just lose their minds. They had no idea what was going on. But as I was scrolling through that the website, I came across this really interesting picture of, it's like poor Anole dead upside down in the snow, somewhere between, where he was coming from and where he was going. He just lost the ability to function, and he kicked it, and I think this is just such a straightforward idea of selection and action, right? And it brings to mind, Bumpus and, so his original studies in response to extreme weather events. And it just so happened that I was studying this particular species, the green anole, at the time this event occurred. I was actually studying the evolution of cold tolerance across the species range. So the green anole originates in Cuba. It landed in peninsular Florida sometime during the Miocene, Pliocene, and then spread out of Florida north and west during the Pleistocene, where it occupies much colder habitats in Tennessee, North Carolina, Oklahoma, where winters are much more extreme than they would experience in their long-term ancestral range in Cuba, certainly. So I was really interested in the evolution of cold tolerance in response to this range expansion of this ancestrally subtropical species. And right before this event hit, I was studying a transect across Texas and Oklahoma, from Brownsville, like the very southern tip of Texas, all the way up to Hodgen, Oklahoma. And this is an interesting transect because, across all the populations I've studied in this species, this represents the most and least cold tolerant populations at the ends of this transect that I have found across the range. And there's this really interesting and sort of beautiful transition of increasing cold tolerance as you move from south to north. So I was in the process of trying to understand metabolic response to cold across these different sites, collecting data on transcriptomes, et cetera, when this event hit. So after this event, it brought up a whole different set of questions, right? So has there been an adaptive physiological response to the polar vortex? If so, what regulatory and genetic mechanisms are involved in that response? And are these responses similar to those governing geographic patterns of local adaptation over longer periods of time? So when we look at this focal winter, this extreme winter with respect to the previous 15 years, we see that at each site along this transect, this focal winter was significantly colder than the winter averages across the previous 15 years. But also, as I said, cold tolerance changes as you move across this transect. So if you look at this in a slightly different way, in this focal winter, the number of days where a population experienced temperatures below its critical thermal minimum, which I had measured prior to the polar vortex, you actually see the intensity of how this event played out, where in the south, the temperatures, during this event, it was much more intense for the southern populations where these organisms are naturally less cold tolerant. So in order to understand physiological response, we use critical thermal method, specifically critical thermal minimum, which is a test of functional performance. So just to give you a view of what this looks like, you have an anole, you tape a thermometer into its cloaca, and you put it in a bucket of ice, and you slowly lower its body temperature by 1 degree Celsius, and then you initiate these riding responses. And the temperature at which an animal is no longer able to right itself after 30 seconds is deemed its critical thermal minimum. So we started this study in 2 different locations, right? So Brownsville, Texas, the southernmost population that was least cold tolerant, and then a population in Austin that that occurred farther north was and is more cold tolerant and was hit less extremely by this winter event. And when we look at critical thermal minimum in the August before these polar vortex storms, you see that obviously Austin, the animals in Austin are able to maintain function at significantly colder temperatures than in Brownsville, Texas. So then after the polar vortex in the spring, April 2014, we went back to these 2 populations and looked at potential shifts in critical thermal minimum. And we see that at the southernmost site where they were hit hardest, you see a significant decrease in critical thermal minimum. So now the survivors of this event are able to maintain function at significantly colder temperatures, whereas you see no significant difference in Austin as our sort of positive control. But this could also be due to plasticity, the fact that they've just come out of winter, it's spring. So we actually went back to these 2 sites in the August of 2014, the same time of year where we got our first measurements, and we remeasured cold tolerance. And we see that still in Brownsville, the survivors of the storm are able to maintain function at significantly colder temperatures. So this seems to actually be selection acting on individual variation in critical thermal minimum at the southern boundary of this species range. When we look across all 5 sites, comparing August of 2013, August 2014, you see that there's a geographic trend. So in the southern part of the range where the animals were least cold tolerant before the storm or hit hardest by the storm, those are the sites where you see the significant decrease in critical thermal minimum. And you don't see that change as you move farther north, where animals become increasingly cold tolerant. So to try to understand, the potential mechanisms, right? Sort of a finer scale understanding of, regulatory response to this event, we use transcriptomics, in this case, liver transcriptomes, to look at patterns of gene expression and regulatory change in response to this polar vortex event. So in order to do this, we used what's called weighted gene co-expression network analysis, or WGCNA, which essentially takes your transcriptome and identifies specific co-regulatory networks, groups of genes that seem to be changing in their expression in a coordinated fashion across individuals. And this is typically how this is represented, but we're going to simplify this a little bit, and we're going to make each of the modules that are identified a circle. All right, so we have our 68 co-regulatory modules that were identified in the liver transcriptome of Anolis carolinensis. Now, these are not all equivalent, so we can look at the size distribution. The smallest is 33 genes. The largest is a bit over 1,000 genes. And across these genes, we looked for those modules whose expression, coordinated expression, was significantly associated with latitude. All right, and we identified 3 such modules, so 2 that are negatively correlated with latitude, and 1 that's positively correlated with latitude. And so given these modules that seem to have like some coordinated expression and association with local environment as you move from south to north, we asked, does there seem to be any sort of systematically biased change in expression in the south that is moving individuals towards regulatory phenotypes that better describe individuals that occur farther north? And when we look across all 3 of these modules, the answer is yes. All right, so you have this disproportionate number of genes within each of these modules that is moving in the direction that you would expect based on the difference in expression between the southernmost and northernmost population. So the regulatory profiles are becoming more northern-like across the boundary of this storm. And again, because we have RNA-seq data, we can actually dig in a level even further and look at patterns of genetic divergence. So here you see patterns of genetic differentiation across the 6 macrochromosomes of the carolinensis genome. And we can summarize this by looking at those regions that show significant elevations in genetic divergence, which you see here in red. There are 8 such regions. And we can compare those with patterns of genetic divergence that differ between the southernmost and northernmost population before the storm, and ask if there are any genomic signatures of selection that seem to overlap between the 2. Right, so you see south versus north in blue here. And then of those 8 regions that we see responding to the polar vortex, 3 of those also seem to respond to latitude of origin. So you do see some similarity in their response. But then we take it a step further and sort of go back to the gene expression data, and we ask, are there any genes that are differentially expressed across the boundary of the storm that also sit under any of these FST peaks? And we identify several differentially expressed genes under those peaks, most of them concentrated in chromosome 2. And these genes represent different aspects of cholinesterase activity and sodium symporter activity, which are different mechanistic associations with neurological function, which we know is a very thermally sensitive mechanism in ectotherms. Right. So in that case, right, we have sort of lay out the functional component of this temporal genomics, the idea of like not just looking at the sequence level, but using regulatory data, if you have it, to understand, functional response, like physiological response to these sorts of extreme weather events. But if you have the right data, you can also ask a slightly different question, which is in regards to the repeatability of evolution, right, in response to these sort of extreme weather events, right? So we'll call this parallel temporal genomics, for what it's worth. And this is actually a new project that I've been doing in collaboration with Colin Donahue, who is at Brown right now. So Colin in 2018 published a study in Nature looking at populations of Anolis scriptus on water-K and pine-K in Turks and Caicos before and after Hurricane Irma and Maria. So Hurricane Irma and Maria sort of passed through this region, like one right after the other, and sort of devastated much of the Caribbean, including these islands on the Turks and Caicos. And he found that, in Anolis scriptus, that the survivors of these hurricane events actually showed significant changes in their limb proportions, presumably in association with clinging ability. So some of these aspects of limb proportions increased and some decreased. So for instance, humerus, length of the humerus increased, length of the femur decreased, the hind limb longest toe decreased, and then the forelimb and hind limb toe pads increased. And all of these are presumably related to aspects of clinging performance in association with hurricane survival. All right, so in collaboration with Colin, basically, we've taken genetic samples from both of these Ks, water K and pine K, before and after this hurricane season. And we've done RAD-seq analyses of these two populations. And we see that, first of all, there seems to be perfect population structure between these two Ks, despite the fact that you could probably walk across the boundary that separates the two. But this is interesting because it actually gives us a means to look at parallelism, or like the repeatability of response to these extreme weather events in these two populations. So the first thing that we did, because in this case, I think we had about 180 individuals, is, we tried to identify genes that were involved in individual variation in each of these limb components that responded to this hurricane event. And so just to give you a view of sort of like what this looks like. So here is an Anolis scriptus, and it's simulating these hurricane force winds. And you see it's holding on for dear life, trying its best, and its back legs sort of pick up first. And then eventually, he's really holding on there. He's really holding on there. But eventually he lets go. But as you can see, when you're talking about the reduction in size of the hind limbs, the hind limbs have a tendency to pick up first, and as they perch, they sort of like kind of stick out, so kind of like the edges of a kite. So if you have shorter limbs, then it like decreases the wind resistance, presumably here. But the point is, so we have each of these characteristics, and we know the directionality of change, and we have the individual variation amongst all these individuals. So we can identify, potential QTL associated with each of these traits. And then we have, we actually use the population branch statistic to identify genetic change that is distinct to these post-storm lineages, right? And we can actually look at the overlap between these two in order to identify strong candidates, strong genomic candidates for signatures of selection. First, we looked for regions that show parallel response between both of these islands. So, you know, in black here, you have regions of chromosome 2 and chromosome 3 that responded in that seemed to show genetic divergence in the post-hurricane lineage in one island, but not the other. But in red, you have those that actually show this parallel change, this lineage-specific genetic divergence across both of the islands. These red regions account for 4 different genes, and each of these genes have some known function in musculoskeletal formation. So TNP01, which is a bone mass regulator, connectin 3, which is associated with tendon formation and musculoskeletal tissues, and PDZN3, which negatively regulates BMP2 induced osteoblast differentiation through inhibition of Wnt signaling. So each of these has to do with like some aspect of skeletal muscular formation. But of these genes that show this parallel response, we were only able to identify one that also showed a significant correlation with any of these traits, and that is HS6ST1, right, which is a heparin sulfate sulfotransferase. And this is actually the top candidate associated with the reduction in the length of the longest toe. So Zani in 2000, you know, he described this relationship between toe length and clinging performance, showing that decreases in toe length actually correlate with increases in clinging performance on rough substrates. And we know that in response to this storm, this species decreased the size of their longest rear toe. And this gene we know is involved in limb bud development. So knockout experiments in chicken, for instance, resulted in truncation of the developing limb. And mice that are deficient in this gene display reduced or missing hind limb tarsus or phalanges. And you can see sort of CRISPR knockout mouse strain here. So if you look at focus in on D and E, you can see, so D is sort of the wild type, right? And you can see the formation of the different phalanges. But then in the knockout, you see extreme reduction of this phalanges formation. So now we're in the process, you know, we have this sort of functional relationship between this gene and the trait that we know that the gene is seems to be associated with. But in order to do a direct validation of function in anolis, we are actually teaming up with Doug Minke to perform CRISPR knockout of this gene specifically in anoles to see how it affects skeletal morphological development in this species. Okay, so now we have, we went through the temporal functional genomics, we have the parallel temporal genomics, each of which I think reveals something very interesting, about the process of rapid evolution or response to selection. And but both require some degree of luck, right? Like you have to have the data beforehand, the genetic data beforehand, or the transcriptomic data beforehand, in order to compare it to the data afterwards. But that's not always the case. Obviously we don't always have that data. It'd be great if we did, but we just, even as scientists, we don't have that kind of forethought all the time. And sometimes these events are very hard to predict. But when you're considering the full range of temporal genomics, sometimes the temporal and the genomics don't necessarily have to walk hand in hand, in the sense that you can have temporal data and genomics to actually reveal biological response to rapid events. And I think that, maybe this is playing a little hard and loose with, with temporal genomics, but I still think it fits this trend. And I'm going to give you an example of this that's actually quite different from these previous two examples. So we know, temporal data have been really important for our understanding of selection. Elton and Haldane in 1942, they used this this time series data from the Canadian fur trade to give us one of the first actual estimates of selection in the wild. And this is like selection against the silver morph of fox coat colors, because that silver morph was more desirable in the fur trade. Basically they tracked over time, over a period of about 100 years, how the frequency of that morph and the cross morph actually decreased over time, whereas the more sort of standard red morph relatively increased over time. Yeah, and when we're considering hunting mediated selection, this is obviously an issue of human mediated change that has become more and more important as our technology has developed, and as the efficiency and magnitude of hunting have increased for many species around the globe. And this particularly comes to bear in times of civil conflict, where you have military grade weaponry that becomes much more readily abundant in certain areas than would be the case normally. And equipped with that new technology, individuals are more likely to try to take game that they might be more hesitant to otherwise. And that includes large mammals, things like elephants. All right, so this story centers around the Mozambican Civil War, which took place from 1977 to 1992. And during this time, in Gorongosa National Park in Mozambique, the large mammal population was reduced by about 90%, including the African elephant population in Gorongosa, where the population size went from about 2,500 individuals to about 250 individuals over the boundary of this period. And the thing about African elephants is that most African elephants have tusks, but there is a small proportion of females specifically that lack tusks. They're born without tusks and they never grow them. And during this time, there's also a significant increase in the frequency of tuskless females in this population. So typically this occurs at about 2% in a population. In Gorongosa before the war, it actually occurred at about 18.5%. And that's likely because there was, the war with the Portuguese happened directly before that. But during these times of civil war, ivory is used to fuel arms purchasing for these conflicts. So just to give you an example of sort of what I mean when we talk about tusklessness, I mean complete tusklessness. So in these pictures here, on the left you see the skull of a bull, and if you look, into those tusk cavities, you can see that they go all the way up into the skull. But on the right, you see the skull of a tuskless female, and you can see that those tusk cavities are completely ossified over. So there's no room for tooth growth whatsoever. But we know that these tusks play an important role for this species. And after the Civil War, we notice that there's an increase in the frequency of that tusklessness from 18.5% to almost 50%, or a little over 50% of the female survivors of this war were completely tuskless. And then in the generation born after the war, a little over 32% of the females were completely tuskless. And in this case, right, we have, like, this has always been told, I think, as sort of a just so story of evolutionary response. But in actuality, we have these two things that are playing out simultaneously over time, right? We have this change in phenotype frequency, but we also have this significant decrease in population size. So it's possible that the change that we're seeing is just a function of an extreme bottleneck event, right? Sort of random sampling from an extreme bottleneck event. So what we did with this temporal data is we simulated this population size change with the known proportion, the known tusk phenotype proportions before and after the war. And we asked, how likely is it that you could see this magnitude of change if there was equal survivorship probability across these different morph types? Right. And, using these maximum likelihood methods, we estimate that tuskless individuals had to survive at a little over 5 times the rate of their tusk counterparts during this 15-year period in order to see the magnitude of change that we see in this population. And that is even given, this extreme reduction in population size. So in this case, right, the temporal data show us, they very strongly point to selection as a major factor in causing this change in the tuskless phenotype. All right. And given the sort of odd inheritance, the fact that this trait is female specific, many times traits that are female specific have a tendency to be X-linked, and when they're only expressed in females, that has a tendency to be associated with male lethality. So the question is it possible that the reason why we don't see tuskless males is because this trait is actually X-linked dominant and male lethal? So we didn't have a proper pedigree from this population, but we did have calving survey information. So we had phenotypic data from mothers and their offspring, which we could use to test this hypothesis of a single locus X-linked dominant male lethal trait. So first, we would expect that under this model, 2 tusked females should be homozygous for the recessive state of this trait, meaning that 2 tusked females should always have 2 tusked daughters. And they should also always have 2 tusked sons as well. Whereas tuskless females should have offspring that have a variety of phenotypes from completely tusked to completely tuskless. And when we look at the maternal phenotype and the phenotypes of their offspring, we see that 2 tusked females, their daughters are 2 tusked, whereas tuskless females, their daughters, they can either be 2 tusked, they can be completely tuskless, or they can also have a unilateral phenotype, an intermediate phenotype, having either one only the right or only the left tusk. Now, this intermediate phenotype is kind of tricky because individuals can also lose their tusks, just through through fighting or through stripping. So in a conservative, so we take the conservative approach. If we assume that all of this variation is genetic, then we can come up with some sort of estimation of the proportion of data that is explained by this single locus model. The second part of this model has to do with the tuskless females, right? So if this is X-linked dominant and male lethal, you'd always expect the completely tuskless trait to be apparent in the heterozygous form. Right. And because every male is always carrying the recessive allele, you'd expect that half of a tuskless female's offspring will be affected by this trait. So half should be tuskless, half should be tusked. But if it's male lethal, then the half of those males that would be affected, we should never see, which means that we would expect a female bias in the offspring of tuskless females specifically. And when we specifically 2/3 of their female, 2/3 of their offspring should be female. And when we look at the percent of female offspring, we see that amongst the tuskless mothers, they do show a significant female bias that is within 1% of that expectation under complete male lethality. And but when we look at the two-tusked females in Gorongosa, it's not significantly different than the 50-50 sex ratio that we would expect normally. And we can also compare that to Amboseli National Park, which has long-term data. And we know that tusklessness occurs at very low frequencies, at about that 2% that I mentioned before. And they also don't show any significant variation away from from that 50-50 ratio of offspring. So this suggests that that this female bias skew in offspring is specific to the tuskless morph in Gorongosa. So altogether, just a little bit over 87% of the survey data support this inheritance pattern for tusklessness in Gorongosa. But in order to actually identify the targets of selection, that is sort of where the genomics come in, right? So we have this temporal data that lays out this story of selection, but the question is, what has been the genomic target of that selection? In order to do that, you have to actually go out and get DNA from elephants, which I can tell you as a lizard biologist is a very, very different thing to try to do. It requires two teams, one in the air, one on the ground, and they have to sedate the elephant. They collar her to track her movements. When we take blood samples, then they reverse the sedation and everybody runs for their life because nobody's trying to be around when that girl gets back on her feet. But this is the means by which we got these blood samples. So we were able to take blood samples from matriarchs in Gorongosa, both tusked and tuskless matriarchs, and use them to try to identify these potential targets of selection and loci of large effect associated with this tusklessness. And in order to do this, just to sort of summarize this, because we have this hypothesis of it being actually an X-linked dominant male lethal trait, we combined a series of tests that would where we would expect patterns that are specific to selection on this type of a trait, right? So you have your standard genetic divergence between your tusk morphs measured through DXY and FST, increases in linkage disequilibrium, which you'd expect as a function of selection. But also because, again, this is a trait, if it's X-linked dominant and male lethal, it would have to occur in the heterozygous state. You'd also expect that the locus underpinning the trait to show an increase in heterozygosity. So at the intersection of outliers between these four statistics, we found 305 windows. Of those 305 windows, 8 of those windows actually occur on the X chromosome, again, which you'd expect if it was X-linked dominant and male lethal. Those 8 windows represent 2 loci, but only one of those loci actually has a known function in mammalian tooth development. And that is ameliogenin X. So across this region of the X chromosome, we see that, we see this increase in heterozygosity, linkage disequilibrium, and genetic divergence between our tusk morphs. Now, what's interesting about this region is that AMELX also lies within this tight bit of the X chromosome that is also surrounded by genes that are known to be associated with male lethality. And we also know that in humans, deletions in this region are associated with ameliogenesis imperfecta, which is a rare sporadic X-linked condition in humans that that's usually lethal in males, so disproportionately affects females, and this is caused by a deletion in this same region. Even more interesting is that ameliogenesis imperfecta presents with a syndrome of craniofacial abnormalities, but amongst them is either microdonty or complete agenesis of maxillary lateral incisors, which in elephants are their tusks. All right, so in this, we have this combination of temporal data, and we have this genomic data that sort of point to human-mediated selection on a single gene or a few genes within the species genome. But now we actually have an opportunity to understand how human-mediated selection on a few regions of a single species genome could potentially have cascading effects across entire ecosystems. All right, so we know that tusks are multi-purpose tools that are used amongst other things for like stripping bark, which allows these animals to eat woody vegetation. And our preliminary data suggests that there's actually differences in diet between tusked and tuskless morphs, where these tuskless individuals are eating significantly less woody vegetation and more grasses. Now, if you consider the role that that elephants play in the savanna ecosystems, right, this change in behavior could have a lot of downstream cascading consequences, right? So, if they're not stripping bark, are they still knocking over trees, so that they can get to that bark? Well, this is something that we're trying to understand. But those differences in habitat modification can also lead to differences in nutrient distribution, as well as differences in niche creation and resource availability for other species, which can affect species diversity and abundance, and then ultimately ecosystem health and sustainability. All right, so this combination in this case of temporal data and genomics begins, I think this really interesting series of connections where we can look at human-mediated changes on a single gene or a few genes in a single species, and link that all the way to wholesale changes in ecosystem function. All right, so in summary, the diverse utility of temporal genomics, we can use it to understand physiological response to rapid environmental change, the repeatability of response to selection, and also the cascading ecological impacts of human-mediated selection. So I think that, again, right, temporal genomics writ large offers a really interesting tool to help us to link evolution and anthropogenic change, so helping us to reveal the lasting biological impacts of anthropogenic change through evolution, and then providing fundamental and unique insights about the process of evolution through our understanding of anthropogenic change. So with that, I'll say thank you so much, and I'm happy to take any questions that you might have.
Cool, that was great. Let's give Shane a virtual hand here. Yeah, and if anyone has any questions, please, if you can raise your hand or you can post a question in the chat. If you want, I can call on you to read that question, or I could just read that question myself. Yeah, and I'm also gonna post our website in the chat now too, because I neglected to do that earlier. But yeah, thanks Shane. That was fantastic.
Awesome, thank you.
I can maybe start off with just something that was going through my mind in that in your last section. What are there any prospects for getting temporal genomic data from maybe tusks that were taken from these elephants, or is it too hard to find where those ended up?
So there certainly is a possibility. I think the major hurdle is logistical. Although there was just a paper, I think, published in Current Biology by Al Roca's group at Illinois, sort of tracing the origins of ivory that was from, I think, the oldest known ship sunken off of the west coast of Africa, and they were actually able to take DNA from those ivory samples to identify geographic origin, number of herds, et cetera, et cetera. So there's definitely a lot of I think possibilities there. I think it's just mostly logistical, like trying to figure out where these samples exist and what sort of metadata exist along with those samples.
Cool. Thanks. Malin's got a question. Malin, do you want to unmute yourself and ask?
Sure. Yeah, thanks, Shane. That was a really cool talk.
Awesome.
across all three different stories. I had a question about sort of maybe the longer term temporal consequences. You had especially the first two, really interesting examples of selection from a single event.
Yeah.
And then I was, trying to think, what do you think happened? These events have return times.
Mm-hmm.
Do you think these are populations that are actually evolving back in the interim? And then this is something that's going to be happening periodically. And then, some of these events may actually get more common. So I was just wondering if you could speculate about how these play out over those longer timescales.
Yeah, so that's an interesting question. I should say, as of yet, we don't have the empirical data to directly get at that question. But there are a few things. Like one, when you're talking about selection at this time frame, right? I mean, it pretty much has to be soft sweeps, right? Selection on standing genetic variation, and as such, it's unless the event is incredibly strong, I wouldn't necessarily expect any of the targets of selection to actually sweep to fixation, which means that, under relaxed periods of selection, I would expect fluctuations back down into sort of intermediate frequencies that represent, that would represent the traits being selected for. I should say that I am in the process of planning the very early stages of planning a collaboration with a couple of labs in Australia. I actually went out there a couple Christmases ago during the recurrent heat waves and fires that are going on in Australia. And, they have longer term temporal data across these recurrent heat waves from Australian fruit bats that have died during these events. So by looking at the individual, by sampling DNA from the individuals that we know sort of died during each of these recurrent events and comparing that against background genetic variation, across that same time frame, we can get at this sort of temporal repeatability of selection, if the same regions are being selected for again and again, or if there's sort of idiosyncratic response, and then sort of what happens in the meantime.
Really interesting.
That's great. We have a question from Sabrina in the chat, and then we have another hand raised. But Sabrina, do you want to ask that question, or I can just read it off too?
Oh, I can do that. Okay. I was wondering if there is a work going on investigating the population history of the tuskless elephant using genome-wide variation to see if the selection is also affecting the autosomal part of the elephant, because I think you only do it for the sex chromosome. Am I right? Yeah.
So the data that we have, it is whole genome sequencing data. And the part that I showed, basically, we're testing hypotheses based on the model of inheritance, but we are also doing broader scans of selection across the entire genome to identify other potential loci that may be the target of selection. Because while there does seem to be this sex-linked locus of large effect, there's also variation that remains unexplained by that single locus model, suggesting that there are other loci that are involved, and they may be sex-linked or they may be autosomal, but the only way to really try to identify them is to do these genome-wide scans, which we're doing right now.
Oh, cool, thanks.
Thanks, Shane. So Mark, you've got a question, you've got a hand raised. You can unmute yourself and ask.
Yeah, totally fantastic talk. And what strikes me as I think reassuring with this work is I think a lot of it is usually on these large population size, high heterozygosity species that experience some event. But my assumption is that these lizards don't have huge population sizes, yet still fairly substantial variation is maintained to kind of persist. So could you maybe just provide some intuition about maybe some population size numbers and heterozygosity? And then just a little bit similarly, I also assume when you're doing these GWAS, it differs in that you can't like get thousands of individuals to do the traits, yet you're still detecting a signal. But maybe some of the lack of signal at some genes, do you think is due to underpowered GWAS? It's totally challenging, but
yeah. So let's see. So there are two parts of that. So in terms of anoles, right? So green anoles, it across the entire species range, which is like all of the Southeast, essentially like everywhere from Texas, Oklahoma, all the way to the East Coast. Everything outside of Florida is essentially one panmictic population. So it's actually gargantuan population. Anolis scriptus, on the other hand, even like on those tiny little islands, like you could walk, from the end of one island all the way to the end of another island in, I don't know, an hour probably. That's like trudging through the water. So like pretty limited space. And we still even see those signatures of selection in that case. Of course, the most extreme would be the elephants, right? I mean, these are individuals that occur on the complete opposite side of the spectrum in terms of population size, generation time, amongst all the animals on the planet. You would expect, these types of animals would be, would really get the crap end of the stick when it comes to like having to respond rapidly to, to human mediated selection. Yet still, even in that case, and amidst an extreme population decline, you still see, these signatures of selective response. So, I definitely think the efficiency of selection, obviously, will change with effective population size and standing genetic variation. But in some cases, even on the other extreme, you still see these signatures. What was the second part? Oh, the GWAS.
Do you think you're constrained by like the number of individuals or?
Absolutely. Oh yeah.
Okay.
Oh God, yes. There's no way it's not constrained by the number of individuals. So in this case, we I think in the hurricane study, we had about 180 individuals. Which is it's a nice number for like non-model systems. But in terms of GWAS, it don't go with a whole lot of juice, right? Mm-hmm. And so what we've done is essentially we've taken a very conservative approach in trying to identify the most likely targets of selection, right? In the sense that, in order to qualify as a candidate of selection, a locus would have to display branch-specific signatures along the post-hurricane lineages in both islands. So they'd have to display that in parallel, and also a significant association with a phenotype that we know responded to that storm or to that series of hurricanes. And in that case, there was only one locus that met all of those criteria. But even the other genes that we see that show this parallel signature that don't map specifically onto one of the traits, that could either be one due to limited power of the GWAS with the sample sizes we had, or because of their link with, other traits that may be important, but that were not directly measured. For instance, one of those genes, we know that, genetic variation or like knockout of that gene results in like decreased grip strength in mice, for instance, and that is not something that was directly measured, in this study. So yes, the short answer after the long answer is yes, it's definitely limited. And I think that in those cases, you have to take a pretty conservative approach, to gain some confidence in the outcome of the analysis.
Thank you very much.
Yeah. Great. Yeah. It's fantastic. And I don't know, it's noon now, Shane. I don't know, or it's noon Eastern time now. I don't know if you have to, if you have places to go, but I had one just, final, maybe concluding question, because we were getting a bit into methods, and a big focus of this seminar series is best practices. Do you have any wisdom that you can give to people after having done a couple of these studies, like things that you would do over again, things that knowing now, from these studies that you would change, or, any wisdom for people who are interested in doing this kind of work?
I think if I've learned anything, from doing these types of studies, so like I always wish I had long-term data. Like after the polar vortex, study, like it was a great study, but if I had continued, like if I had the ability to like keep going out, year after year to collect the data, there's, a whole different set of, of questions that I'd be able to get at, and the same, and you see the utility of that long-term data, even when it's not genomic data with the elephant project. I mean, that survey data, goes back to, I think like the 60s or so. And without that long-term survey data, even like our understanding of, the effects of, bottleneck event versus selection on this trait, it would have been impossible to disentangle, even if we were able to identify a gene responsible for the trait. The context wouldn't be there. So I guess the biggest bit of advice, I would say is like, if you have a system that's related to any of this sort of stuff, that even has the potential, be vigilant about data collection, because you just never know when it's going to come in handy. And quite frankly, it's more likely that it's going to come in handy as we move forward. So if you can, definitely go for it.
Cool. Okay, thank you. Yeah, that's super useful. All right, well, thanks everybody for showing up, for attending, and we have another talk April 22nd, so 2 weeks from now, from Evelyn Jensen on Galapagos tortoises. So hopefully everybody can rejoin us then. Thanks everybody for coming.
Thank you. Take care.
Yep, you too, Shane.